Hello and welcome to the talk through of this paper. Hopefully we're okay on number one. Yeah, just make sure you can never say sugar. It's always reducing sugar or non-reducing sugar, so be careful on that. But there's nothing really to do there. 1.2. Um, yeah, I know it's not on the mark scheme, but I always say start with a fresh sample of sugar. It wouldn't work if you've just done a Benedict test on one solution. It's not going to work doing it again. But yeah, you need a fresh sample of sugar. Boil it in acid, neutralize it, and then heat with Benedict's reagent. And then you get your colour change. That proves it's now a reducing sugar. It became a reducing sugar when we boiled it in acid. It hydrolyzed the glycosidic bond to go from a disaccharide to a monosaccharide, and all monosaccharides are reducing sugars. A lot of people struggle on this when it was in the exam because people didn't know what an element was, or just put CHO as well. So yeah, always it says name the chemical elements. So yeah, it's amazing how many people get that wrong. Hopefully that wasn't you. And then we're on to tissue fluid and stuff. Yeah, so people got a common mistake on X is to put blood, but yeah, it's the fluid. And therefore, yeah, it's blood plasma. So put plasma there. And tissue fluid is straightforward. Yeah, describe one way. Look how I've underlined one. Just it's easier to make me focus on it. So yeah, I've gone for plasma proteins. Are only found in the plasma as they are too large to be filtered out. It's a bit like the kidney, and you're probably onto the kidney soon. So you can just use that knowledge, and then explain how fluid leaves the capillary at the arterial end. Yeah, it's going between the cells. I mean, just to show you quickly, if that's my capillary, and you can see the cells are all touching apart from at the arterial end, and the venous end is over here. The pressure is so high it kind of forces blood plasma. Sorry, it doesn't force blood plasma. It forces small molecules between, like water, between these cells here. It pushes the cells apart to form like a little pore. So yes, so it's called ultrafiltration. Very similar to what happens in the kidney. So yeah, and it just points out that yeah, it's the high hydrostatic pressure. So basically, water pressure causes small molecules to be forced between the endothelial cells of the capillary wall. Gets me 2 marks. Oops. On to this one. It says with reference to Fick's law. So I always write out Fick's law. Rate of diffusion is proportional to the concentration gradient times surface area divided by thickness of membrane, but nothing's changing here apart from the activity of the muscle. I've got an active muscle or a resting muscle. So yeah, so it can only affect the concentration gradient as I've put there. And then as active muscle cells will be using oxygen in aerobic respiration. I put now, that it's the terminal electron acceptor, just because if it was on a year on a paper 3, this could could be a marking point. If it's on paper 1, it's based on like topics 1 to 4, so it wouldn't be on a mark scheme. But again, there's no harm in putting it, and therefore there's more. So there's the one in the active is using more oxygen in aerobic respiration. Therefore, there's a greater or steeper concentration gradient for oxygen to diffuse into those cells. So that's just anything with any kind of like gas exchange or anything like that. Like this one is get down Fick's law and then relate your answer to the bits of Fick's law. Then we've got a tangent to do. I always do this. If I've got a partially numbered scale, I've popped on the other numbers. Yeah, this y-axis starts at 10.5, and that's critical because I've put my tangent on at 50, and it's going from, and you get a lot of leeway where this goes. But yeah, the y difference is 12.5 minus 10.5, and my x difference is 1. I actually put that in over my delta. Oops, can't even write it. Try again. Delta x equals that. So yes, my gradient is 140. Again, it's only awkward because of the scale. Again, the number of people who forget to who think every y-axis starts at 0 is quite high. So yeah, Again, do like I've done and pop some lines on the graph to make sure you know what it's going up in, so you get an accurate measure from it. You don't need to revise hard for questions like that. You just need to be better or have a technique. Question 3, it's showing this precursor molecule and then one that carries proline, right? So it's proline, that comes up later in the question. Which bonds is just hydrogen, that's straightforward. Describe, right, not explain, just describe how the precursor molecule is modified to produce a tRNA molecule specific. Well, the grey bits have gone, and it's got this ACCOH added on. So I just said that, yeah, the shaded nucleotides are removed by hydrolyzing phosphodester bonds. I know I'm explaining a bit here, but you never know fully what's on the mark scheme. And then add nucleotides with adenine, cytosine as their bases or ligated on, forming phosphodester bonds. It should say by forming phosphodester bonds on that. Ligated is it's a DNA ligase does it to DNA. This is RNA, but yeah, joining bits onto an established bit of nucleotide is called ligation, which you might have met when you've been doing your recombinant plasmids. Oops, right, big scroll, sorry. Move on to this one. So yeah, so we're carrying on with our proline, and yeah, that told me on the previous on that diagram. I'm not going to scroll back, but GGU was the anticodon of the tRNA, and it carried proline. And I always put these in order, so I've got my DNA, my RNA, and my tRNA. So I've got that from the question, my GGU. Then I go back to mRNA and I go to DNA, and then by doing it, I've answered the question as well. But they're going to ask that. Classic 3.5, just multiply by 3. 3 bases code for, it can't be become or are, they code for amino acid. It's a really common misconception where people think that bases are amino acids. They are not. 3.6 is just knowledge. Explain, again, tells me it's knowledge. So yeah, introns are removed from pre-mRNA during splicing, and mRNA is only made of exons. Again, I'm saying two things. I'm not quite sure what's exactly on the mark scheme, so I'm mentioning introns and exons in case it wants me to refer to both. I'm mentioning pre-mRNA and mRNA in case they're on there, and I'm naming the process of splicing. 3.7, just don't write transcription, it's translation. So protein synthesis, Golgi apparatus, not Golgi, full stop. And then this is quite a tricky one. Shows the segments of mRNA, right, and the amino acids coded. Describe the single mutation in the DNA, right, that resulted in the new amino acids shown. So we've swapped, so it was glycine, it's now serine. Well, how do I know the base sequence for serine? Because again, in the question above, it was proline. Oh, I've got serine here, so I know AGC codes for serine. So yeah, so that G must have been replaced by an A. So that's gone from an A. So it's going to an A. And therefore, yeah, it's a substitution of the fourth base, the G, to an A. However, in the DNA, well, it would be cytosine coding for the guanine, and it will be a thymine coding for the adenine. So yeah, cytosine is replaced by thymine, and you can't put C replaced by T. You have to always name the bases. Number 4, which is just showing our fatty acids. Yeah, so if we're joining, it's condensation, and ester is fats. How can I make it more liquid? Well, you can either make it shorter or saturated, or unsaturated. So it's 2 marks, so I'm going for the more detailed. Yeah, that carbon-carbon double bonds are present in the fatty acid tails to cause unsaturated fatty acids. I put fatty acids there to change that to a T. There you go. And then it's 2 marks. I might get 2 marks for this, I'm not sure, but I can also say that the hydrocarbon tails of the fatty acids could also be shorter. Both of those cause it to be more fluid and have a lower melting point. And a diagram of how one fatty acid above would bind to glycerol. So yeah, just pop the ester bond on, and I can put an R instead of all those C's and H's. But yeah, all we are doing is I'm forming water. So off comes water, H2O, and I join what's left. So I've got a C left there that's going to join onto that C. I've lost that OH, so that's gone, and then my double bond O sticks up at a neater angle. And then number 5 is meiosis. So yeah, it's quite hard to get wrong. Is 5.1 because it's I've got a nucleus present, and then the nucleus is now the chromosomes are condensing. I've lost the nucleolus as well. Then they're going to go to it's going from 3 to 2. Then they align on the equator. Then they go to number 1, and then they're going to go over here. So it's 3, 2, 4, 1, 5. P is the nuclear envelope, the allowed nucleus. And Q is the centromere. I think Lake Windermere. Lakes are like little puddles of water, circular, just like that. So that's my something lake mere. And therefore, it's centromere. Whereas a centriole, they look more like square, rectangular things. So yeah, people get confused with centrioles and centromeres because they interact. But yeah, it's a centromere. And then 5.3, with reference to the cell numbered 4. Well, if we look at cell number 4, all right, it's doing some crossing over, even though it really happens in prophase. Some crossing over there, and on like either side of the equator. So I can talk about, yes, independent segregation is shown where maternal and paternal copy align randomly above and below the equator. And also non-sister chromatids of homologous chromosomes form a bivalent. Again, that's when they come together where they associate. It's bivalent. Chiasma are the crossover points, and the same length of DNA. So alleles of the same genes are exchanged between the maternal and paternal chromosome, and that process is called crossing over. Oh, we're done. That's the first 5 for this 5 tranche. So I shall stop there because these videos are boring, or these borings are videos. So I shall end.